Welcome to our lecture on animal biotechnology. Today, we are going to explore the fascinating and essential world of cloning vectors. If you have ever wondered how scientists actually move DNA from one organism to another to create life-saving medicines or study complex genetic diseases, you are in the exact right place. Let us dive in and build our understanding from the ground up. To start our journey, I want you to imagine that you have just invented a brilliant new recipe for a life-saving medicine, but you only have one single copy written on a tiny piece of paper. If you want a massive factory to start producing this medicine, you cannot just throw the paper over the factory wall and hope for the best. You need a secure delivery truck to carry your recipe inside, protect it from being destroyed, and hand it directly to the factory managers. In the microscopic world of biotechnology, a cloning vector acts exactly like that delivery truck for your DNA. Now that we have our delivery truck analogy in mind, let us look at the formal scientific definition. A cloning vector is a small, highly stable DNA molecule, which is very often derived from naturally occurring plasmids or viruses. Its primary job is to act as a specialized vehicle to artificially carry foreign genetic material into another living biological factory, such as a bacterial or animal cell. Once it is safely inside the host cell, the vector ensures that the foreign DNA is replicated. This means it is copied alongside the host cell's own genetic material, preventing the host from recognizing the naked DNA as foreign garbage and destroying it. For this microscopic delivery truck to function properly and do its job effectively, it needs three critical anatomical features. First is the origin of replication, often abbreviated as ORI. This is a specific DNA sequence that tells the host cell's machinery to start copying the vector. Second, it requires a multiple cloning site, which acts like a molecular zipper containing unique recognition sites where we can insert our foreign gene. Finally, it must have a selectable marker, such as an antibiotic resistance gene. This marker is crucial because it allows scientists to easily identify and select only the cells that have successfully taken up the vector. Let us visualize what we just discussed to make it perfectly clear. On your screen is a schematic diagram of a standard plasmid cloning vector. Notice the circular structure of the DNA? The green region represents the origin of replication, ensuring the plasmid can be copied by the host. The blue region is the multiple cloning site, and you can see where the gene of interest has been inserted into it. Finally, the red section is the selectable marker, specifically an ampicillin resistance gene. This simple but elegant structure is the absolute foundation of genetic engineering. Having seen what a vector looks like, you might wonder why they are so incredibly important to science. The invention and refinement of cloning vectors completely revolutionized modern medicine, agriculture, and biological research. Without these microscopic delivery vehicles, we simply would not be able to produce life-saving recombinant therapeutics, like human insulin or blood clotting factors, on a massive commercial scale. Furthermore, they are absolutely essential for creating genetically modified animals and developing advanced gene therapies, where healthy genes are delivered directly into patients to cure devastating genetic diseases. Now, the actual laboratory process of using a cloning vector to deliver DNA into a host cell is called transformation. However, unfortunately, this process is never perfectly efficient. Not every single cell in our test tube will successfully take up the vector. Therefore, we need a rigorous mathematical way to measure how successful our delivery trucks were at getting inside the factories. This measurement is known as transformation efficiency. It specifically tells us how many successful host cells, or transformants, we produced per microgram of vector DNA used. By calculating this, scientists can determine if their delivery system is working well. This brings us to the mathematical representation of our experimental success. The formula for transformation efficiency is T equals N divided by M multiplied by VT divided by VP. Conceptually, this formula simply scales up the number of colonies you actually counted to figure out how many you would have gotten if you plated the entire volume. Let us break down the symbols. T is the transformation efficiency. N stands for the number of successful colonies counted on the Petri dish. The symbol M represents the mass of vector DNA used, measured in micrograms. VT is the total volume of the cell suspension in microliters, and VP is the specific volume actually plated. If the number of colonies N increases, your efficiency T increases, meaning a highly successful delivery. Let us see this formula in action with a practical laboratory scenario. Suppose a student uses 0.05 micrograms of DNA. After transformation, the total volume is 1,000 microliters, but they only plate a small sample of 100 microliters. The next day, they count exactly 250 glowing bacterial colonies. We need to calculate the transformation efficiency. We plug these known values into our formula. First, we divide 250 by 0.05 to get 5,000. Then, we divide the total volume of 1,000 by the plated volume of 100 to get a scaling factor of 10. Multiplying 5,000 by 10 gives us our final answer of 50,000. Physically, this means that for every single microgram of this specific vector, we can expect to successfully engineer 50,000 bacterial cells. Moving beyond the delivery of single genes, another critical mathematical application of vectors is building what we call a genomic library. A genomic library is a massive collection of thousands of host cells, where each individual cell contains a vector carrying a different tiny fragment of an organism's entire DNA. Because we are chopping up a massive genome into tiny pieces and randomly inserting them into vectors, we face a statistical problem. We need to know exactly how many total vector clones we must create to guarantee that every single piece of the genetic puzzle is represented, without missing the specific gene we are looking for. To calculate the exact number of vector clones required, scientists use the famous Clark-Carbon formula, which relies heavily on the laws of probability. The formula is N equals the natural logarithm of 1 minus p divided by the natural logarithm of 1 minus f. Let us understand these variables. N represents the total number of vector clones required to build a complete library. The symbol P stands for your desired probability that any specific gene is present, usually set very high, such as 99%. The variable F represents the fractional size of the genome contained in a single vector insert. If you want a higher probability P, the required number of clones N increases dramatically because achieving absolute certainty requires vastly more samples. Let us apply the Clark-Carbon formula to a realistic library construction problem. Imagine we have a newly discovered animal with a total genome size of 4 million base pairs. Our standard cloning vector can only hold an insert of 4,000 base pairs. We want to be 99% confident that every gene is present. First, we find the fractional size f by dividing the insert capacity of 4,000 by the total genome of 4 million, giving us 0.001. Next, we substitute our probability p as 0.99 and f as 0.001 into the formula. The numerator becomes the natural logarithm of 0.01, which evaluates to negative 4.605. The denominator becomes the natural logarithm of 0.999, which is negative 0.001005. Dividing these two values gives approximately 4602.7. Since we cannot have a fraction of a living cell, we must round up. The final answer is 4,603 clones. Physically, the researcher must create and store at least 4,603 individual colonies to be 99% certain the entire genome is captured. We have covered a lot of ground today, so let us summarize our key takeaways. First, we learned that cloning vectors are the indispensable delivery trucks of modern biotechnology, allowing us to safely transport, protect, and replicate foreign DNA inside host cells. Second, we saw how to use the transformation efficiency formula to mathematically measure the success of our DNA delivery process. Finally, we used the Clark-Carbon formula to calculate exactly how many clones are needed to build a massive genomic library. By mastering how these molecular trucks operate and the mathematics behind them, you are unlocking the ability to push the boundaries of genetic research. Great job today!